What if I told you that there were evolution-proof drugs and not killing the bacteria could be our solution to the antibiotics crisis? Every time we take one of these antibiotics, that drug tries to kill every bacterial cell it can find, whether that's by interfering with the cell wall, by interfering with its metabolism, by messing up how the bacteria makes proteins, by affecting how it reads and copies DNA, or by making the bacterial membrane leaky. Each class of antibiotics is a family of drugs that all affect the same target, but all of these targets are specific to bacteria, which is what makes antibiotics magic bullets. So we can take them, they will kill the bacterial cells, but they won't even affect our human cells. But just like humans, bacterial cells are individuals, and errors that happen when they're copying their DNA cause mutations, and by chance, some of those mutations means that the antibiotic can no longer kill the cell. And we say that the bacteria has become resistant. Antibiotic resistance is a totally natural process. This is just evolution. We discovered most of our antibiotic drugs from fungi and other bacteria in the soil, which release these antimicrobial chemicals to keep themselves healthy, and we just exploited that. But because this is happening in the environment all the time, there are resistant strains of bacteria in the environment all the time anyway. And the oldest ones we've found date back 30,000 years.
An antibiotic-resistant superbug.
Threat of antibiotic-resistant bugs.
Antibiotic-resistant infection.
Superbugs. Antibiotica.
Antibiotics.
Deviennent de plus en plus résistantes.
Resistant pandemic.
The end of the antibiotics era.
And as big a danger as climate change or warfare.
If this process is natural, then what is the problem? Well, just like humans, bacteria don't want to die, and there is a very strong incentive to evolve resistance when your life is on the line. And every single time we give an antibiotic to a disease, we are putting extra pressure on that disease to evolve so it will evolve faster. Because every single time you take an antibiotic, you are killing millions, if not billions, of bacterial cells. And that is what we call, in the field of evolution, a jolly big selection pressure indeed. And so if you've got one or two cells that have by chance mutated to become resistant, they're not only going to survive, they're going to flourish because you have wiped out all of their competition. And every time you take an antibiotic, you're educating the disease against our arsenal, our weapons, and giving them a chance to evolve. But what's worse is that bacteria have this nifty trick of being able to swap genes, not only between different individuals of the same species, but between species. And that means that a disease doesn't have to have experienced an antibiotic. We don't have to have given that particular class of antibiotics to that particular disease before for it to be resistant. No, they can just talk to another species of bacteria that causes another disease that we've already given that antibiotic to, and they can swap notes, and now you've got diseases in this world that are resistant to every single class of antibiotics that we have discovered. We no longer have any weapons that are effective against them. Like I said, resistance is a natural process. It's just evolution in action. And it's been happening in the wild for millennia. So why is it a problem now? Well, unfortunately, there are two factors that together are creating a perfect storm. First, we are misusing the antibiotics that we already have, and two, we're not coming up with new ones. When we think about what is causing the antibiotics crisis, we often think about some poor, overworked doctor who is being browbeaten by their patient into prescribing antibiotics for something like a flu, even though the doctor knows that antibiotics do not work on viruses. Okay? Or maybe there is a patient who's been prescribed antibiotics for their bacterial disease properly, but then they start feeling better and so they don't finish taking their course of pills and they just kind of give up halfway through. And those are big problems. But what shocked me the most while researching this video is that globally, 73% of our antibiotics are given not to humans, but to farm animals. And this isn't a case of some well-meaning farmer saying, oh, Daisy's looking a bit peaky today, better call the vet and see if she needs some medicine. No, we're talking about large-scale farms and corporations routinely giving all of their animals antibiotics all of the time just to make them grow a bit faster. In fact, the industry has renamed antibiotics growth promoters because they're happy to just, reduce the efficacy of our most vital medicines just for a few extra % of profit. And it's not like we even need to do this because in the EU, since 2006, we have banned the use of antibiotics as growth promoters. We do not have the routine use of antibiotics in producing meat in the EU, and yet we are still able to produce meat at competitive prices. I don't know if you can tell, but this makes me incredibly angry because it's not like diseases stay in animals. If you pump a pig full of antibiotics, you'll get pig diseases that are resistant to antibiotics. And if that pig disease becomes a human disease, well then we are screwed. If we're just chucking around all of these antibiotics with reckless abandon and giving bacteria every chance they need to become resistant to them, surely, at the very least, we must be coming up with new drugs that bacteria have never seen before and so can't be resistant to yet. This is a timeline of when each new class of antibiotics was brought to market. Now, because drugs in a class all act in roughly the same way, if bacteria become resistant to one member of the class, it's very easy for them to be resistant to the whole class. And this is when we first found a disease that was resistant to each class of antibiotics. This is a period we refer to as the golden age of antibiotics, when we thought we could rid the world of bacterial disease forever. But you can see that from 1970 onwards, pharmaceutical companies just gave up researching new antibiotics. They were making iterations of other classes of drugs, but these are the only 3 new classes of antibiotics that have been brought to market since the 1970s. And all 3 of them were discovered in labs before 1990. If we, don't want to end up in a post-antibiotic age where something as simple as a paper cut or strep throat or a tooth infection can kill you, then we need to slow down the rate at which bacteria evolve resistance. But evolution is an inevitable consequence of life. And if we're fighting evolution, that is a battle we are never going to win. But what if we could design evolution-proof drugs? That is, drugs where it's not only hard for bacteria to evolve resistance, but the bacteria don't want to even try to evolve resistance. And that is the concept behind antivirulence drugs.
My name is Stefan Sieber. I'm a chemist working on bacteria.
What is virulence?
Virulence is basically the reason why bacteria are so dangerous, at least the pathogenic bacteria, because they produce toxins and they have a versatile toolset of toxins that have many deleterious effects to our human cells. So we have many bacteria in us, in our gut, in our skin, and most of them are harmless. They are not a problem. And there are even some pathogenic I probably also have Staphylococcus aureus on my skin, but usually it doesn't make a problem because our immune system can keep them basically calm. But once they reach and grow to a certain density, they can monitor their community, and then they start producing these virulence factors. Only at a point, and this is so nasty, when they realize that they have a chance to overcome the defense.
So they're like, oh, it's our moment. Now we can attack the body.
Exactly. Yeah. So that's what they do. That makes them so nasty. And for a healthy person, that's not a problem. But for weak persons with immune system problems, it becomes a challenge.
And one of the big things I hear about antivirulence drugs is evolution-proof antibiotics. So
Exactly.
Are antivirulence drugs actually evolution-proof?
It's difficult to say because there are no drugs yet on the market. So I think first of all, we have to see it on the market to see how it behaves.
I know it can be hard to get your head around at first, but when you've got a bacterial infection, it's not the fact that there are bacteria inside your body that causes the disease. It's when those bacteria then go on to produce virulence factors, these chemicals that attack our own cells. That is what makes us ill, not just the mere presence of bacteria. And as Professor Sieber points out, we are so early on in the research of antivirulence drugs, we're not even at clinical trials yet. But if we turn to the field of evolutionary theory, which, kind of my thing, kind of what I have a PhD in, then we can see a few ways, at least, in which it's possible to make drugs that are evolution-proof. The first easy target of evolution-proof drugs are the bacteria that never meant to be harmful in the first place. It doesn't help them to produce these virulence factors. So maybe the bacteria is adapted to life in a pig and it's ended up in a human by mistake, or it's adapted to life on our skin and then has ended up in our blood by mistake. And it never meant to kill the human officer. And so if we have a drug that stops them producing these chemicals, because it's not harming them, they're not going to resist. It's not like we're killing them. They're just like, oh yeah, sure, we didn't need that anyway. There's another theoretical target for evolution-proof drugs, and it is so clever, but incredibly difficult to explain succinctly. Not that will stop me giving it a go. So bacteria are social. They produce chemicals that they can communicate with each other, and they secrete these chemicals into the environment, but it costs them to do so. It costs energy and food to produce them. And so it's like everyone paying the cost in an office of paying money into a kitty jar, but then everyone gets to benefit from there being free biscuits in the office at all times. Hooray! But now, if you have a drug that stops everybody from paying money into the jar, sure, all the bacteria are now to disadvantage. Nobody is getting free biscuits. But, and here's the genius bit, any resistant mutant would be a bacteria that is now paying money into the jar. So yes, there is a small benefit of there being biscuits around, there being virulence factor around, but everybody is getting that benefit and only the mutant is paying the costs. And that just can't evolve. That will be exploited. Oh, it's so, genius. Can you tell me about the work that you've been doing personally in this kind of field of research?
We are also working in the field of antivirulence by addressing one protein in the cell that is responsible for the regulation of virulence. And if you block this protein, it cannot activate this transcription of or translation also of the virulence genes into the toxins.
So you've got this one switch that basically controls the rest of the bacteria to turn it from being a kind of harmless, it's just doing its thing inside your body, to full-on attack mode.
This is one major switch that is regulating a lot of virulence factors. Yeah, so we were the lab that discovered the inhibitors against the switch.
I'm Ulrich Betz. I'm the Vice President of Innovation for MAG.
So can you tell me what was the idea behind the Future Insight Prize?
The purpose of the Future Insight Prize is to honor and enable groundbreaking research in areas of key importance for humanity. That's multi-drug resistance. And we're awarding this this year in 2020 to Professor Stefan Sieber.
And you get €1 million, which is an amazing amount of money. Congratulations. What are you going to do with it?
Much. Yeah, so we have a lot of plans. So we have quite a lot of data, and we have quite a lot of compounds. We have even done screening of compounds. Now we really need an expert that makes more sense out of this data. Then we would like to extend our work into different bacteria. So there's a lot of bacteria that need different growth conditions, for instance, anaerobic, so they are not growing under air. And then, of course, what's really important to see really if it's working in a preclinical setting, and this is very expensive.
I want to say a big thank you to Merck for funding this video as part of their Curiosity initiative and for, supporting smaller science creators like me. I'm an evolutionary biologist, and so I find the concept of evolution-proof drugs fascinating. But it's only one of the solutions to the antibiotics crisis. There's also going to be faster testing. There's an idea called soil hotels where we culture the unculturable, and so we can bring them into a lab. And on Merck's Curiosity website, they have a whole page dedicated to all of this new research. It's really well written, and they've got links to all of the papers as well if you want to do future reading. So the link will be in the description, and I really recommend you find out more there.